I am Dr. C.SWAMINATHAN, Assistant Professor in the Department of Microbiology, St.Joseph College of Arts and Science, Cuddalore. In this video, we are going to discuss about Staphylococcus aureus. Properties of the bacteria. Staphylococcus aureus is a gram-positive cocci. They are arranged in grape-like clusters. They are non-motile, non-spore forming and non-capsulated. Some strains possess capsule. They are aerobes and facultative anaerobic organism. When the organism is cultivated on neutron agar, organism forms the golden yellow color colonies. Pathogenesis and clinical features. Staph aureus causes infections and intoxications in human being. Now let us discuss about the infections. Skin and soft tissue infections caused by Staph aureus are furuncle, paronychia, folliculitis, cellulitis, wound infection and breast abscess. Furuncle is the boil. Paronychia is the infection of tissues adjacent to the nail. Folliculitis is the inflammation of the hair follicle. Cellulitis is the deep infection of the skin. Breast abscess is the accumulation of pus in the breast. Musculoskeletal infections are arthritis, pyomyositis and osteomyelitis. Arthritis is the inflammation of the joint. Pyomyositis is the purulent infection of the skeletal muscle. Osteomyelitis refers to the bone infection. Central nervous system infections are abscess and meningitis. Respiratory infections caused by Staph aureus are tonsillitis, pharyngitis, sinusitis, otitis and lung abscess. Tonsillitis is the inflammation of the tonsils. Pharyngitis is the inflammation of the pharynx. Sinusitis is the inflammation of the sinus. Otitis is the inflammation of the ear. Lung abscess is the accumulation of pus in the lung. Endovascular infections caused by Staph aureus are bacteremia and endocarditis. Bacteremia is the presence of bacteria in the bloodstream. Endocarditis is the inflammation of the endocardium. Next was the intoxications. Under this topic, we are going to discuss about the staphylococcal food poisoning. So, if we consume the food material which is containing the enterotoxin that is produced by the Staph aureus, we will get the staphylococcal food poisoning. The clinical features of staphylococcal food poisoning are nausea, vomiting and diarrhea. Nausea is the vomiting sensation. Toxic shock syndrome, which is shortly known as the TSS. TSS is caused by the TSST1, which is produced by Staph aureus. TSST is the toxic shock syndrome toxin 1. Clinical features of toxic shock syndrome are low BP, fever, diarrhea and skin rash. Next one is the staphylococcal scalar skin syndrome. Staphylococcal scalar skin syndrome is caused by the exfoliative toxins A and B, which is produced by the Staph aureus. The clinical picture here is the outer layer of the epidermis gets separated from the underlying tissues. Next one is the laboratory diagnosis. For the laboratory diagnosis, specimens such as pus, blood, CSF, urine, sputum and fecal matter can be collected from the patients. Microscopy examination. Gram staining of the clinical material reveals the gram-positive cocci in clusters. Culture. For the culture, specimen is inoculated on the blood agar and incubated overnight. Colonies will be surrounded by the beta type of hemolysis. Specimens from the heavily contaminated sources such as vomitus and fecal material are inoculated on the mannitol salt agar medium. On mannitol salt agar medium, Staph aureus forms yellow color colonies. That yellow zone is due to the fermentation of the mannitol. Biochemical test. Staph aureus is catalase positive, oxidase negative. Staph aureus ferments the mannitol with the production of the acid. Novobiosin sensitivity test. Novobiosin sensitivity test is carried out by means of placing novobiosin disc on the Muller-Hinton agar, which is already seeded with the test bacterial isolate. So, if it is Staph aureus, it will be novobiosin sensitive. I mean, the zone of incubation will be more than or equal to 16 millimeter in size. If it is a Staphylococcus saprophyticus, it will be novoboicin resistant. Staph aureus will be novoboicin sensitive. Staph saprophyticus will be novoboicin resistant. Tube coagulase. Tube coagulase test detects the free coagulase enzyme. For carrying out this, carrying out this test, 0.1 ml of the broth culture is added to the 0.5 ml of human serum in a narrow test tube. The tubes are incubated in a water bath at 37 degrees Celsius for 3 to 6 hours. If the organism is coagulase positive, plasma clots and does not flow when the tube is tilted. Thank you very much for watching this video. Thank you.